Revolution.
Welcome to Genomics Revolution. This is Brad Goodner from the 2019 Hiram College Genetics course, hosting this episode on the genomes of two strains of the genus Xenarabdis. Xenarabdis nematophila, ATCC 19061, and Xenarabdis bovieni, SS 2004. I will call them X nematophila and X bovieni from here on out. These strains are in the bacteria division Gamma proteobacteria and within the family Enterobacteriaceae, meaning they are closely related to well-known human gut symbionts and pathogens such as E. coli and Salmonella enterica. However, Xenarabdis strains do not live inside the guts of humans. Rather, they are gut symbionts of particular soil nematodes of the genus Stenorobnema. The bacteria are needed for the maturation of the nematodes. Not only that, the bacteria and the nematode host work together to kill certain insect larvae and use them as nutrient sources. Wow, that is a complex interaction. And that is why a group of labs from the US and Europe, including my lab at Hiram College, came together to sequence these two genomes. One of the biggest problems we faced with these two genomes is that they have lots of repetitive sequences in them, including transposable elements. This makes it very hard to assemble sequencing reads into the full genome. Think about it. If the same 400 base pair sequence is present 10 times in the genome, then how do we know which fragments go together? This is rarely an issue for a genome in bacteria or archaea, but it gave us a lot of headaches for these two particular Xenarabdis genomes. To get around the problem, we worked with a company in Madison, Wisconsin called OpGen Technologies to use a genome-wide restriction mapping technique called optical mapping. You very, very gently lyse bacterial cells onto the surface of an electron microscopy grid. Cut the DNA as it lays on the grid with a particular restriction enzyme, and figure out the length and order of the fragments. This strategy was critical for getting these genomes done. The two Xenarabdis genomes each have a single circular chromosome, ranging in size from 4.26 to 4.43 million base pairs. X nematophila has an additional plasmid of 155,000 base pairs. The two genomes encode between 4250 and 4350 proteins. The Xenarabdis genomes were compared to two previously sequenced genomes from a closely related genus, Photorhabdis, that lives in the guts of nematodes of a different genus, that being Heterorhabditis. The two bacterial genera inhabit different parts of the nematode gut, and the two nematode bacterial symbioses attack different insect larval hosts. Comparison of the genomes across the two genera showed that there are many orthologous genes in common, some of which probably deal with basic metabolism, but also some that deal with living inside a nematode. That said, these shared orthologs only account for about half of any one genome. On the other side of the coin, the two Xenarabdis strains had many genes in common not found in the Photorhabdis strains, and vice versa. This means that the two closely related genera of bacteria do some really different things. For example, each genus appears to make some unique insect-killing and antimicrobial peptides and other molecules. Moreover, we also found that the two genera solve the same ecological problems, such as dealing with oxidative stress, using different strategies. These two genera diverged from a common ancestor that might have lived inside nematodes, but they have converged on solving certain ecological problems from different angles. Since the publication of these genomes, several labs have tested hypotheses using wild-type and mutant strains in both in vitro and in vivo experiments. For example, Singh et al. Used mutants of X nematophila unable to make different antimicrobial compounds to figure out how the bacterium kills off competitor bacteria inside a dead insect larva. Murphin et al. Figured out that X. bovieni helps its host nematode outcompete related non-host nematodes. Bacteria helping a roundworm kill a caterpillar so that each can benefit, even if there are competing bacteria and nematodes around. Who would have guessed that? Thanks for listening.